Welcome to BioMindscape. Today we are going to know about the synthetic biology. Synthetic biology is a science of designing and constructing new biological part, devices and system or reprogramming the existing one for the beneficial purpose. What beneficial purpose? In various fields like healthcare, agriculture, environment, energy and industry. In healthcare, their application is used in designing microbes to produce vaccine, insulin, or cancer therapy, anti-malarial drug by engineered yeast, artemisinin production. In agriculture, it is helping the creating the drought resistant, pesticide resistant crop and biofertilizer. In environment, biosensor to detect the pollutant, engineered bacteria to clean the oil spills, bioremediation, in that also it is important. Then in energy, microbial energy to produce the biofuels or hydrogen, in that it is important. In industry, green manufacturing of chemical like plastic or detergent using the microbes, in that it have an important role. So synthetic biology, we are going to know a structure of the mycoplasma mycoids, the first synthetic cell created by the Craig Venter and its team using a chemical synthesized genome of the mycoplasma mycoid. And it is this is main important example. We are going to know today the structure of it, and other than that, it is also synthesized in Yeast Genome Project. It is important in entire yeast chromosome is being synthesized by it. Then CRISPR-engineered organism, it is used in medicines and agriculture for targeted modification. In India is investigating in synthetic biology through DBT initiative and National Mission of Biodiversity and Human Well-being support researchers in an area. And then there are various in short, we are going to know about the structure first. We have known the application of synthetic biology in the various sectors like healthcare, agriculture, environment, energy, industries. So, unlike traditional genetic engineering, which modify existing genes, synthetic biology built entire genetic circuits or organism from the scratch, like engineer built machine using standard parts, like that. Like making the gene, ribosome, promoter, then using or designing new genetic circuits. Gene synthesis is writing the DNA sequence digitally and assembling them in the lab. Genome editing tools like CRISPR-Cas, then ZFNs and TALENs are allowed in precising the DNA modification. Systemic modeling like using computational models to design or predict the biological behavior. Then pathway engineer like modifying or creating new metabolic pathway in microbe to produce a drug or fuel or chemical, and that it is important. So it is an interdisciplinary field that combines the principle of biological engineering, genetic and biotechnology, computer science to design, construct and redesign the biological system of an organism. So it aims to create a new life or modify the existing one by synthesizing the genetic material, DNA or RNA, or by creating artificial cell and programming the biological system to perform a specific task. So let's know the structure of this. So this is a formation of synthetic mycoplasma cell using a homologous, recombinant and synthetic DNA. So this process is major milestone in synthetic biology, where the scientists create life form from chemical synthesized genome. So the formation of synthetic mycoplasm, here we will know step by step how the synthetic genome is assembled and used to create a synthetic cell. So we will know what is a synthetic cell. So first step is transform in the yeast. So synthetic DNA fragments of a mycoplasma mycoids genome are made, and each fragment have a overlapping end, same color homology to allow the recombinant. So these fragments are introduced in the yeast, which has a powerful recombinant machinery. Then inside the yeast nucleus, the DNA fragment assemble into one complete synthetic chromosome. We can see here, this is a homologous chromosome, recombinant joining the overlapping ends of each other, fragment into a full mycoplasm genome. Then the assembled synthetic genome is extracted from the yeast. Here we can see it, that is a transfer the synthetic chromosome in the mycoplasm. It is then transplanted into the recipient cell, that is the mycoplasm. A related species that has its own DNA removed. Then the result is that the cell contains two chromosomes. It's original and the other is synthetic. So one is the M capricolum gene, original one. Synthetic is M mycoids. So during cell division, these chromosomes are randomly stored into the daughter cell. Then isolation of the synthetic mycoplasm. After multiple division, some cell will have only the synthetic chromosome. So these cells are now called synthetic mycoplasm. It behaves like M mycoplasm mycoids but entirely based on a man-made genome. So here is a reporter gene, blue-white colony selection. A reporter gene LAGZ was inserted in the synthetic genome, absent in natural M capricolum. So when the plate on the exgal media, here the blue colonies synthesize the mycoplasm where the LAGZ is present, and the white colonies are the wild type where the M capricorn, where there is no LAGZ. So no LAGZ is seen. So if the LAGZ is absent, no blue colonies will be occurring. So this is simple color test to identify the synthetic genome function. So synthetic biology enable the creation of artificial genome from a scratch. Yeast is used as a host for genome assembly by homologous recombination. The functional validity of synthetic cell can be done using the reporter gene like LAGZ, and this process can lead to the synthetic life form controlling the entire human design DNA. So it is an interdisciplinary field which is helping to design, construct and redesign biological system of an organism for various purposes and various sectors, for synthesizing the genetic material DNA or RNA and creating artificial cell, and programming the biological system to perform specific tasks. It can build entire genetic circuit organism from a scratch, like an engineer building a machine using a standard part. watching. Do like, share and subscribe.